Well, welcome to the second panel, which is on, and the meeting is on biomarkers. And there's, I think, Alzheimer's disease is a particularly interesting area for biomarkers now, given the data now on Aduhelm and Lilly's compound coming forward. This has been incredibly controversial and therefore interesting, I think, for all of us to talk about. We have several, I think, very distinguished people on the panel today. I'll be co-moderating with Simone Fishburn from BioCentury. Have Al Sandrock, who's the Chief Medical Officer, or was until recently Chief Medical Officer of Biogen and the driver behind Aduhelm. Rob Califf, who's doing heavy duty today, just was on the first panel and now on this panel as well. And I think Rob is well known, the nominee to take over as commissioner of the FDA. Dan Skovronsky is head of R&D at Lilly. And I think Dan is particularly knowledgeable about biomarkers, having developed the PET scan reagents for Alzheimer's disease. So thank you, Dan. Gil Rabinovici is a well-known neurologist, and Mark Duggan is a well-known health economist from Stanford. And then Sue Peschin is here to, I think, represent the patient voice in all of this. So thank you to all the panelists. I would, I actually don't see Al on this oh, there you are. Okay. All right. Good. So let me throw out the first question to Al. And, obviously the approval of Aduhelm has been hugely controversial. And for the time being, let's put away the price, because I think Biogen probably shot itself in the foot with the pricing. And I think, in fairness, Al had nothing to do with that part of it. But there's been so much controversy about this drug, some of it quite virulent and surprising to me. And some of it from people who clearly are not in a position to know the details. And so there's been some sense of jumping on the bandwagon. On the other hand, there are many experienced voices who are skeptical, and the EMA decided against approval. So I think it might be putting Al on the spot here, but maybe you can just lay out a little bit the rationale for approval. Talk about, there are some biomarkers here, right? Obviously, Plaques, Tangles, ARIA can be considered a biomarker. I think it's either biomarkers for an adverse event or for efficacy or both. And so just like to give Al a few minutes to talk about Aduhelm and his views.
Thanks, George. Thanks for inviting me to this meeting again, and I appreciate that the organizers invited me again. Let me start with the rationale for approval as I see it. Number one, Alzheimer's disease is a fatal, progressive neurodegenerative disorder for which there are no treatments that modify the course of the disease. The toll it takes on patients and their families is extraordinary. Number two, the damage to the brain in this disease is irreversible. In other words, once brain function is lost, it's lost permanently. Number three, amyloid plaques and neurofibrillary tangles, as you mentioned, George, are the key neuropathological hallmarks of the disease. In fact, they're the defining features of the disease and distinguish it from other neurodegenerative diseases. So there are these 40 to 42 amino acid long fragments of A beta, which is a protein, which is the main component of amyloid plaques. And human genetics strongly point to A beta as a key causal biological process in the development of Alzheimer's disease, both the early familial forms as well as the late onset sporadic forms. So amyloid plaques are not just any biomarker. It's one of the defining biomarkers of the disease, and it's composed of proteins that are undoubtedly in the causal biological process that leads to disease. So I've heard statements by some people that numerous drugs have targeted A beta, but none of them have worked. Well, these kinds of statements are based on a superficial understanding of the data as I see it. In fact, there's a graph in the clinical pharmacology section of the Aduhelm approval package that shows the data from various dose groups of 6 different anti-amyloid antibodies on amyloid plaque removal as seen by PET scans in patients at various stages of Alzheimer's disease. And there's a strong relationship in the graph between the degree of plaque removal and efficacy on the clinical dementia rating scale. The first generation anti-amyloid antibodies did not remove very much amyloid, and they had little, if any, effect on clinical outcomes. It's only the more recent antibodies, Aduhelm being the first of several, that targets the forms of A beta that are enriched in amyloid plaques and thus have more robust effects on amyloid removal. And they are the ones that show dose and time-dependent efficacy on clinical outcomes in controlled clinical trials. Senior FDA officials have said in published statements that they determined that Aduhelm is reasonably likely to predict clinical benefit of the drug based on the removal of amyloid plaques. FDA and Biogen have acknowledged that some uncertainty on the clinical benefit of Aduhelm exists based on the partially discordant results of the 2 phase 3 trials, and Biogen is committed to running a confirmatory trial. In fact, it's required to running a confirmatory trial in a timely manner to reduce the residual uncertainty. So the data on Aduhelm's efficacy are forthcoming. The key question now is not whether further study is needed. The key question is whether or not the drug should be made available for use by patients in the early stages of Alzheimer's disease, a fatal, progressive, irreversible disease, while the studies are ongoing, or should we ask patients to wait until the confirmatory study is completed? It'll take about 4 years. I heard the panel discussion previously on the timely completion of confirmatory trials. Biogen has estimated it'll take about 4 years to complete the confirmatory trial after it is initiated, and the initiation is planned for March of this year. 4 years is based on Biogen's experience with setting up hundreds of sites in the phase 3 program, rebuilding the soccer stadium, to use the analogy from the previous trial, time required for screening patients, reasonable enrollment rates, and an 18-month trial duration, which is how long it takes to determine whether the drug can affect the course of this progressive disease. So every year, hundreds of thousands of Americans will progress from MCI, or mild cognitive impairment, where they can still lead independent lives, to later stages of Alzheimer's disease where they're functionally impaired, and they will no longer be eligible for treatment based on the current label. If it's reasonably likely that a clinical benefit can be achieved, isn't it in the American public's best interest to make the drug available now, prior to study completion? So the statute says reasonably likely to predict the clinical benefit is what's required for accelerated approval. It doesn't say it's certain to predict a clinical benefit. So I believe that accelerated approval pathway was established for just this type of situation. And I think the FDA had the best interest of the American public in mind when they approved Aduhelm by accelerated approval in June of last year. So I'll end by answering the second part of your question, which is, how do we know that this was the right decision? The additional trial will obviously settle the question about Aduhelm's clinical benefit, at least that's the hope. But we may not have to wait that long to determine whether the rationale for approval was sound, because there are 2 phase 3 trials that are going to read out later this year with similar anti-amyloid antibodies that target the form of A beta in the plaques. These have already been shown to robustly remove amyloid plaques, and the results of these studies may go a long way towards settling the issue.
Okay, thank you, Al. Look, I think that's a great start. I don't think this session should be focused around was that decision the right one or not. I think the session should be focused around removal of plaque, reduction of tangles, how those correlate with, let's say, slowing of the decline on cognitive tests, and what are reasonably likely to predict success in AD. I think that's a great start. And Simone, turn it over to you.
Yeah, so I'm going to bridge between the last session on accelerated approval and where we are now and biomarkers. Actually, even before the last session, I think it was Peter Marks who talked about the importance of public faith in science, something we know that is costing us dearly in terms of the pandemic. In this situation, especially regarding the biomarker, even leaving aside the actual decision, you actually have a divided scientific community, which I refer to as believers and non-believers. But, amyloid has probably been the biggest flashpoint. And I'm going to ask Rob what you think the controversy over the acceptance of amyloid as a surrogate endpoint means for accelerated approvals in Alzheimer's, and whether you think that ultimately this might have a positive or a negative, I'm assuming it's not going to have zero impact, impact on future use of the pathway.
Well, Simone, as you know, when we made up the program for this meeting, it was not even in my state of consciousness that I might be going back to FDA. So I'm going to not give anything like my usual charged up participation in a panel. I'll just make a couple of quick points. I completely agree with your point that we have to build public confidence in the science. And at least right now, I think we're really at a loss about how to do that societally. And it's just evidenced by what's happening in the pandemic. So you're bringing it up is important. Secondly, by definition, in accelerated approval, as we discussed in the last panel, the surrogates that are used are unproven. They're not validated surrogates. That's in the definition of accelerated approval. And so it's a judgment call by the FDA. And then thirdly, I would just say, if there is a consensus in the scientific community, it will go better. And so, and lastly, I'll just point out that 5 years ago, we started something called BEST, which is a collaboration between the FDA and NIH to try to bring rational coherence to this issue of biomarkers and intermediate endpoints, to make sure that when we throw the terms around, that we talk about the same things. And I worry that somehow that effort has gotten drowned out by all the commotion, because I think if we're clear about where we're uncertain and we couch the scientific debate in a different way, it probably would do better in terms of helping the public understand that we are dealing with uncertainty here. And I think that that's something we really have to get back to, because there's a lot at stake well beyond Alzheimer's.
Right. Yeah, if we just want to expand on that thought a bit, because some of the biomarkers, for example, plaque removal, might be a good biomarker at some stages of the disease and not a good biomarker at other stages of it. Could be a good biomarker early, not a good biomarker late for efficacy. So there's a temporal component to all of this as well. So maybe Dan, you have, Lilly has a antibody in phase 3. Maybe you can comment on that a bit. And maybe on the relationship between the onset of the disease, the timing of drug intervention, how early can you intervene, when is it too late to intervene, and maybe talk about the temporal components and how we should think about the markers in that frame.
Yeah, thank you, George, and thanks for inviting me to be on this panel. What an exciting time to be working in Alzheimer's disease, thanks to hard work from so many people, including several of my co-panelists here. maybe just a second to congratulate Al Sandrock on all of his accomplishments across a long career at Biogen, working on so many different brain diseases. Thank you and congratulations, Al. I point out that scientific advances are no stranger to controversy, and it's great to be on a panel on a controversial topic. So on to donanemab, which is our antibody, and like aducanumab, it's a very potent plaque-clearing antibody. There are a few key differences to the molecule and how we ran our trials. I think the first is around data, and this is important. We have unconfounded clinical data from a RCT. We had a successful phase 2 clinical trial. It hit its primary endpoint, its statistical significance, at full population. That sounds like a small thing. It's a really big thing. It's the only time that's ever happened, actually, in Alzheimer's disease. Every other trial has got a but or a if or almost to it. It's important in the context of accelerated approval. I think accelerated approval probably works best when you have a combination of clinical outcome data and biomarker data. And so I think, as Al was saying, we have now clinical outcome data, at least from donanemab in phase 2. We have one of the phase 3 trials from aducanumab, and we have earlier and less robust data, I think, from the other agents. And so, I think our clinical data package hopefully helped to make the case for accelerated approval for aducanumab as well as the whole class. But we still need more data, and we have confirmatory clinical trial data coming from our phase 3 trial next year. So the window here for accelerated approval without that kind of confirmatory data, at least for donanemab, is quite short. Again, consistent with all the discussions we've had on accelerated approval and the need to get data quickly so that we can hopefully build public confidence in a new therapy, or if it doesn't work, then withdraw it from the market. And the second important comment here on donanemab comes to the biomarker effects and the surrogate outcome itself of plaque lowering. With donanemab, we see very rapid and deep plaque clearance. We hypothesize that could make a difference. I think if plaque clearing is the surrogate, if you believe that's correct, then it stands to reason that getting more people to fully clear faster, et cetera, could have a better effect. And we're testing that directly. We're doing, I think, another important thing in medical research, which is a head-to-head trial of donanemab versus aducanumab, and that's ongoing now. And hopefully we can understand better the relationship of plaque lowering to different outcomes in exactly the same setting. Dosing is critical here, and another difference for us is that we stop dosing once patients get clear of plaques. That means some patients need as few as 6 or 9 or 12 doses to achieve clearance of plaques. That could have benefits here as we calculate the benefit risk of a given treatment or the cost to society, et cetera. The third and final point I make about donanemab, and then I'll come to the timing of plaque deposition, is related to the supportive biomarker data that we've been able to publish and present. I think uniquely here we have tau imaging results on every patient in the trial. And what we were able to see is that clearance of plaque is accompanied by regional slowing of tau deposition in the brain. We can actually see the slowing of tau spread. We have robust and rapid decrease of phospho-tau species in the blood. We've decreased astroglial inflammation as measured by GFAP in the blood. I think tau is a really important surrogate, and even in the absence of amyloid, could probably stand on its own as a surrogate endpoint suitable for accelerated approval. We know people with higher brain tau or even blood tau have worse cognition and also faster progression over time. And on a region-by-region basis, tau levels correlate with patterns of cognitive decline. Gil Rabinovici here has published extraordinary work on that. And so the evidence around tau as a surrogate is quite strong, and once again, bolstering this as a predictive biomarker. I think Gil will probably, as a world expert here on biomarkers, will talk more about that in a few minutes. So on the issue of timing of treatment, George, which you asked me, I think we don't know for sure, but our hypothesis is that earlier will be better for reducing amyloid plaque. We know amyloid deposition is a very early event in the pathogenesis of Alzheimer's disease. So there's a big window to intervene between when a person gets plaques and when they become an Alzheimer's patient and are symptomatic. So we're running a phase 3 trial right now of donanemab in that population, people who have plaques but don't have symptoms yet, to see if removing plaques with a limited course of therapy, just a fixed duration, can stop them from progressing to Alzheimer's. I'm optimistic, but we don't have any clinical outcome data in that population yet. So I contrast that pre-symptomatic population where there's no outcome data yet to the data I just discussed on potential benefits in patients who are already experiencing symptoms. the next few years will be exciting as we see these results.
Yeah.
Gil, all eyes on you now. And I want to take a little bit of a forward-looking view on biomarkers. So Aduhelm requires PET imaging. That is costly. Tau is also, a costly marker to track. So how is it going to stay that way for a very long time? And more specifically, there is a move in the field to blood markers. Some of these have got good correlation with PET. So maybe you can talk about what you think the timeline or likely prospects are for plasma to start standing in as a biomarker.
Thanks, Simone. Yeah, it's really an honor to be on this distinguished panel. I think there are really two parts to your question. One is a biomarker necessary to make decisions about diagnosis and treatment? And the second is, what is the correct biomarker to use? And so just to address the first question, when I went to medical school not that long ago, we were taught that Alzheimer's is really a clinical syndrome, that even though the biology is defined by plaques and tangles, the only way to know for sure if someone has plaques and tangles in their brain is at the time of autopsy. And during life, we just have to give our best guess about whether people have the disease or not. And breakthroughs like Dan's amyloid PET ligand have really changed that landscape entirely. So now as clinicians, we are able, if we suspect that someone may be suffering from early stages of Alzheimer's, to actually have direct biological evidence for the presence of plaques and tangles at an early stage during life. And that's a game changer. It's been a game changer in drug development, and really the ultimate culmination is the accelerated approval of Aduhelm. There are a lot of lines of data that show us that clinical diagnosis is insufficient to really predict who has plaques and tangles in the brain. One study that we conducted was called the IDEAS trial. This was actually a coverage with evidence development trial. That pathway was mentioned by Dr. Tunis in the last panel to determine the clinical utility of amyloid PET. And what we found in that trial was that 36% of people who were seen by dementia specialists and suspected to have Alzheimer's disease had negative amyloid imaging. And so they did not have evidence of amyloid in their brain. So when you think about treatments that are targeting amyloid, you really need that biomarker, or else you're going to unnecessarily treat many, many people. And I think a lot of us in the field were concerned that there was no requirement for a biomarker in the FDA label for Aduhelm, and I would love to see that corrected. I think it is really necessary. So now to move on, if we agree that we need a biomarker for diagnosis, which biomarker can we use? Currently, as clinicians, we really have two options. We have PET imaging for amyloid plaques and also for tau tangles approved by the FDA. And we have CSF measures of soluble monomers of A-beta, phosphorylated tau, total tau. And actually, either CSF or PET are very accurate at determining who has the pathology in the brain, but they're measuring different things. So PET is measuring the fibrillar forms of the aggregated proteins, the plaques and the tangles that the pathologists are looking for. CSF, again, is measuring soluble forms. These are related, they're correlated, but they're not interchangeable. Now, both PET and CSF have obvious disadvantages. PET, as you mentioned, is expensive. It involves radioactivity. CSF involves a lumbar puncture, which is a somewhat invasive procedure. So plasma, of course, has been the holy grail, if you will, of Alzheimer's biomarkers. If we had a blood test, it would be scalable, it would be very accessible, hopefully cost effective. And if you had asked me 5 years ago if this was near, I would have said no way, because we have the blood-brain barrier, we have such low concentrations of these proteins in the blood. There's also peripheral production of these proteins. It's impossible. Well, I was totally wrong, and happy to be wrong about that. We have made tremendous progress in plasma markers of both amyloid beta and phosphorylated tau, and they're looking really, really good. But they're not quite yet ready for prime time. Really, most of the work that's been done has been done in highly selected research cohorts. There's been very limited diversity in the populations that have been studied. And the lab pre-analytics and analytics that have been required to get these results have been pristine. These are really research-grade labs that are able to immediately centrifuge samples, freeze them. We're talking about mass spectrometry or very highly sensitive immunoassays. And so there's still, I think, some work to do for validation and scalability. And as Dan mentioned, the imaging has some advantages that I think plasma will not be able to address. Imaging will tell you how much plaques or tangles are in the brain. It's not clear. Plasma probably can tell you something about that, but it's not as definitive as imaging. Imaging tells you where the proteins are in the brain. This seems to be much more important for tau, where actually the brain regions that are involved are very strongly correlated with symptoms. Plasma, of course, doesn't tell you anything about spatial distribution. And I think for now, if we really need to stage individuals in terms of where they are in the disease, PET has some advantages. And I think more work needs to be done to see if plasma can tell us, for example, if only people who are in early stages of tau spread will respond to anti-amyloid beta monoclonals, is actually our soluble measures of phosphorylated tau as we get in CSF or in plasma, are they going to be accurate enough to predict that staging of the disease and see who is responding? Will plasma be able to tell us who has cleared amyloid for the brain if we really are only going to treat people until their brain is clear of amyloid with a monoclonal and then at least suspend treatment and observe them? So there are a lot of unanswered questions, but as Dan said, incredibly exciting time also to be working in biomarkers, because we are now approaching biomarkers that can really be tested in very large populations in a cost-effective way and will dramatically, I think, change the way that we diagnose and care for patients.
Okay. Obviously, so much, it's such complexity here. I think, if we talk about accelerated approval and then, a post-accelerated approval confirmation, then it takes some responsibility on the part of companies and regulators to do that in a way that makes sense for society and for the healthcare system. So the initial price of Aduhelm, I think $52,000, just threw a lot of oil on the fire, and I think was probably not the best decision that Biogen could have made. It's been recently cut in half, and we'll see if that makes an impact or not. So, Mark, maybe you can talk about whether or not you think the recent reduction in the price changes the view, or how you would think about what would be a reasonable price for drugs like Aduhelm.
Thanks so much, George, for that question. Thanks, everyone, for having me on. It's really an honor to be on with such a great group. And this is hard scientists. I'm a social scientist, as they refer to health economists. And I'm in the middle of sessions at the annual meetings of the American Economic Association, where we're talking about Social Security and unemployment insurance and other things. But a lot of my work over the years has been on the healthcare sector generally, and on pharmaceutical pricing specifically, and the effect of the Medicare program and Medicaid on this. And basically what's happened, what's coming up, I guess this Wednesday, I believe it is, CMS, the CMS decision is something they sort of do all the time for other drugs, whether it's in Part B or Part D. And I think what is typically, they sort of are not thinking a lot about cost in these decisions, because basically it's a decision about, are we going to cover this or not? And then if it's in Part B, we're going to pay basically 106% of the average sales price. And that's our healthcare sector in the US. We have a public sector, Medicare and Medicaid. We have private sector, and these programs to some extent piggyback on what is happening in the private sector. The hard thing here, and the reason we're somewhat in uncharted territory in a way that we have not been before, is that really basically the entire market is Medicare for this. So you can come up with exceptions, obviously, but it's basically well in excess of 95% of the patients who would be likely to take it are likely to be on it. So that makes this this cost consideration, this price consideration harder for CMS, because it's not, they can't just say, oh, well, here's what private insurers are paying for it. We'll pay 106% of that average sales price and go with that. And so I think right now I think that decision to cut it in half certainly will capture the attention of people at CMS. It's captured the attention of a lot of Medicare recipients already. So as you may know, in the current year, Medicare premiums are up 15%. from last year, Medicare Part B premiums for 65 million Medicare recipients. And almost half of this CMS has attributed to just the expectation of the higher spending for this drug. So Medicare recipients are already feeling this somewhat. That's only a fifth of the expected cost because these premiums are only supposed to cover 20% of the cost. So I think the price reduction will loom large, and obviously evidence is still forthcoming. And I commend Biogen and the other companies that are in this orbit that are trying to advance the best available treatments in this space. My read of the evidence is that up to now, the estimates of the sort of monetized, if you try to monetize the health benefits of this treatment, they're still pretty far below $28,000. And I'm sure there are other studies out there, but I know the ICER, Institute for Clinical and Economic Review study, puts it in the sort of neighborhood of $3,000 to maybe $8,000 per year, which is still quite a bit below the $28,000 price. So if we're sort of doing a sort of cost-benefit calculation, that obviously it's a terrible illness and it affects 6 million Americans. Obviously, they've narrowed it too. But I think that this is, I think it will capture their attention, but it's a little hard to predict because this really is uncharted territory. And so I have done research with Fiona Scott Morton at Yale regarding the effect of what we call the Medicare market share on pharmaceutical pricing. In other words, the fraction of customers who are on Medicare, and this drug is going to be the highest ever. And so it's a little bit outside of our experience, but it's going to be big. I will say, even if CMS decides to cover it, and it's not a 0-1 decision. There's nuance here. There can be different amounts of coverage. It's not obvious that providers will use it. I mean, there's certainly some providers who are out there saying pretty loudly that they're not planning to use it, at least not in the short term. But that said, I'm personally very happy that the science in this area is advancing and very hopeful about what the future holds, and want to commend also the people at Biogen for making important advances already, and at Lilly and Roche and elsewhere. So in any case, that's a long-winded way of saying it will, I'm sure it's caught their attention, but there's still a gap between the estimated benefit and this cost.
A lot of points raised, and we'll try and circle back to some of them, but I want to now turn to Sue, who really, representing the patients who are going to be taking Aduhelm or future competitors to Aduhelm. I think we can't get away from this upcoming CMS decision, so very interested to hear how you or the patient community is thinking about that. And I know that you have talked about efforts to weaken accelerated approval. Maybe you want to expand on that and its importance to this debate.
Sure. And Simone, thank you so much for having me. As everybody else said, it's a very exciting panel for me to be on with all of you. So over the past several years, my organization, the Alliance for Aging Research, we have worked on a couple of the national coverage decisions regarding heart valve disease treatments. And Sean Tunis referred to one on TAVR. There's also a separate one on mitral valve disease at CMS. And a lot of these decisions are highly politicized because of the economic impact on payers, specialty providers, hospitals, and health systems. But they're really kind of little league compared to what's going on in Alzheimer's disease most recently. The treatment was highly politicized by being called out by the agency in their 2022 Part B premium notice as one of the main reasons for the increase. And this was really an unprecedented move for CMS, and we believe it was highly inappropriate. And then last week, the current and former chairs of CMS's MACC Advisory Committee published an opinion piece in Bloomberg that was virulently negative about the upcoming coverage decision, and it included a number of misleading points. And so we want to point these issues out when we meet with the agency on the upcoming decision. We support, as an organization, coverage to label for all the FDA-approved monoclonal antibodies that target amyloid for Alzheimer's disease. And we're organizing the Alzheimer's Disease Policy Task Force to pull together the community on this. And we hope that the oncology community will join us, because we have to work together on these efforts, both on these coverage decision issues as well as on accelerated approval. And as everybody here knows, treatments that are approved under accelerated approval require a phase 4 confirmatory trial. And in the case of the first MAB therapy, the sponsor, Biogen, is going to be conducting a long-term extension study and a real-world observational study. All of that said, we're expecting CMS to issue a draft national coverage decision that requires CED. And that's what Sean Tunis talked about. Requiring additional evidence collection through clinical trials or registries under a CMS CED requirement is going to be redundant, and it's going to significantly restrict beneficiary access, including to the other MAB therapies in development. And one thing I think is not being stressed enough is that this coverage decision is going to apply to the entire class of monoclonal antibodies that have been approved, and also those under development. And it's going to cause competition for recruitment, and it's going to prolong getting the evidence results. So why do we need this on top? It's just piling on. The CED requirements in heart valve disease, and Sean mentioned TAVR, and that CED has been going on for 10 years. One of the claims is that these are much shorter and will be much more efficient than the confirmatory trials, and that's just not true. And they've also created a lot of unnecessary hurdles for smaller and rural hospitals, and that exacerbates access issues and disparities for minority and rural populations. And CMS is really, they're very aware of this, and we applaud them for their calling out equity in the NCA on the Alzheimer's treatments. If you look at the TVT registry that Sean mentioned for TAVR, and also applies to mitral valve, the registry only reports 3 to 4% of people of color participating, compared to about 96 to 97% of whites, depending on the year. And I also want to make a quick comment about Dr. Califf's comment about requiring RCTs in the observational studies. We don't think it's a good idea to require that in all these cases. And I think one of the CEDs on mitral valve disease is a good example. With functional mitral regurgitation, guideline-directed medical therapy is the standard of care. But the prognosis among patients that have heart failure and functional MR on guideline-directed medical therapy alone is really poor. And the co-op trial showed that patients that were on guideline medical directed therapy alone died or were hospitalized for heart failure within 2 years, about 2/3 of them in the co-op trial. And the rate of hospitalization was twice as high in the control group, and death from any cause was also almost twice as high in the control group. So given the nature of the intervention for mitral valve repair, it would have been unethical to blind the study. And so CMS recognized this in the CED that they put out, and we want to make sure that that is still allowable for future CEDs. We have broader concerns regarding the utilization of CEDs. CED falls under CMS's NCD statutory authority. It was created by the agency. It wasn't created by Congress. So there's no real enforcement mechanism to ensure timely research reporting compliance. So there's really like an ad hoc process that leaves Medicare beneficiaries in a state of uncertainty regarding their treatment. And you see it all across the country. There's real variability right now in coverage for this new Alzheimer's therapy. And oftentimes the MACs are prolonging the process out and all of that. So we're supporting the Novel Act, which is sponsored by Senators Burr, Bennett, Carper, and Scott, that would give CMS unique CED statutory authority and require the agency to clarify what specific evidence is needed to close a CED and to provide a timeline for collecting evidence. The other issue that Sean mentioned in passing was the effort to push adjustable rebates for accelerated approval treatments. And this was an idea that was pushed by ICER in a policy paper that they put out last year, and they presented to MACC, which is the advisory committee for the Medicaid program. And the folks that support this say that it's about sharing risk with industry and it's going to help patients, but it's really not true. Because in many instances, industry is going to respond by abandoning clinical development for rare diseases, other areas of unmet need. And even ICER said this in the report. They said that the AAP program might dry up entirely, with companies only willing to invest in areas where they can use the regular approval pathway. So we don't think that there should be any kind of a graduated system. We're really worried about that related to UFA later this year in the wraparound legislation. And one last note is just that it's also just sort of a myth that these are draining these programs. Ken Thorpe and Doug Holtz-Eakin did an analysis in the Medicaid program. And at least from 2007 to 2018, the accelerated approval drugs accounted for less than 1% of overall Medicaid spending per year. But they often represent the only treatment options that are available for beneficiaries. So you don't get a lot of juice for the squeeze, except you're going to hurt patients with rare and complex diseases that don't have any other options.
Thanks, Sue. I'm going to turn to Rob, because you brought it up and it's come up before. Going back to real world evidence here, you also talked in the last panel about how the recovery trial in the UK, I think your words were knock the socks off, what was done here in the US. And one of the reasons the recovery trial was so successful was it was really done in, I think, what we call here community hospitals throughout the UK. That kind of system is virtually impossible to imagine here. So and we also know that there were actually relatively few, I think, in the clinical trials, people of color who were included. So what needs to happen? What can FDA do in terms of bringing real world evidence and enabling a much broader reach into sort of outside of just the major medical centers for gathering evidence for this kind of a drug?
Well, I don't think it's hard to imagine at all. In fact, we did it in the 1980s with the large cardiovascular trials that enrolled tens of thousands of people in the US in community settings, and did it in a very simple way that got definitive answers that led to a reduction by 50% in the rate of death from heart attack. And so it's not hard to do. It's just there are many reasons why the clinical trials enterprise in the US has become adorned with all sorts of ornaments which have no practical purpose and don't help get the answer and cost enormous amounts of money. So I think it's actually very fixable. And if we do enroll people where they are seeing community health centers, federally qualified health centers, rural areas, we now have this, what was the tool that allowed us to do in the 1980s? It was a fax machine, because the internet didn't exist for practical purposes. But you can now see a patient, fax in your form, and get the person randomized over the telephone. Not that hard to do. So now we have the internet, and we have a new law and funding from Congress to make the internet available everywhere in the US. So now is the time to do it. But I do want to emphasize the issue with recovery in the UK is I think it did reflect the population of the UK, which is not the same as the population in the US. So if we just did that in the US, we would have a very representative population enrolled.
I just wanted to make one comment, if I may, about this. I think real-world evidence is incredibly important, but Alzheimer's disease, especially in early stages, is quite different from cardiovascular disease or oncology in terms of the outcomes that we're looking at. So when we're looking at changes in cognition and function at early stages, these are harder to measure in a reliable way and in a scalable way across these different settings. This is really quite different than mortality or even hospitalizations or heart events like MI or stroke. And so we really need to think creatively about how to gather this real-world evidence in Alzheimer's disease. Again, in the IDEA study, we asked about the 600 sites that participated across the US to do just the mini mental state exam or the MOCA. These are about 5-minute tests, and we got pushback on doing that even in real world practice. This reflects that there isn't really a standard way to measure these in the community. And so this is just an additional challenge, I think, something that we can solve, but will require creative solutions, in particular in this disease, in particular in early stages of this disease.
Yeah,
I agree. I would just say that you can do these things now, as you just mentioned, because we do have access to people 24/7. We just have to figure out how to do it.
Yeah, I agree, Gil, with what you and Rob are saying. I think there's a way to do this that's not quite real world yet, but in RCTs, we're trying to get out to where the patients are, taking vans to set up study sites in church parking lots and get a more diverse and community-based sample, and then use centralized raters, to do the very detailed cognitive assessments and things like that. It's a new frontier, but it's definitely worth pursuing, I think.
I just have a question for Rob, Gil, and Dan on that, or anyone else. How do you think about monetizing so suppose you accurately estimate it, which is hard, it's a formidable challenge for all the reasons that were just mentioned. And then now we want to convert that into a dollar value. Curious how you think about that. I'm a health economist, so I think about that issue a lot.
Estimate what?
What? Estimate the dollar value of the improvement in cognition or the slower rate of cognition. I'm curious. I'm a health economist on a panel with a lot of hard scientists. I'm curious to hear how we think about that.
I mean, again, Gil made an important point that outcomes like cognition are more subtle and complex. And, I often say being a cardiologist is relatively easy because it's not too hard to tell whether someone's dead or alive. Although I have seen 2 people declare dead in my clinical career in intensive care units who were not. So sometimes you get it wrong, but not very often. I would just say, heck, in those trials I mentioned in the 1980s, if you measure the outcomes that matter to people in a clinical trial, the methods for evaluating those outcomes, as you well know, are known. It's an evolving science. The FDA is very involved in it. But the key is you got to do the trials in a way that actually measure the things that matter to people in a way that you can compare the intervention group to the non-intervention group without bias. And then I think it's pretty straight. I would argue it's pretty straightforward. In cognition, we may argue about the value, but that's why the scientific enterprise needs to focus on this.
The issue is an interesting one. I do believe a lot revolves around clinical measurement. If you want hard endpoints, such as time to nursing home, or time to the inability to lead independent lives, you need a lot of time because many of these diseases are slowly progressive. Or you need more sensitive outcomes that can read out in a shorter period of time, but those tend to be, you need good measurements that are reliable and done by people who know what they're doing. One hope, I think, here is the use of devices such as the iPhone or computers, which may quantify change, more subtle changes that can be measured in a shorter period of time. But I agree with Gil and in some ways Rob and Dan that this is going to require a lot of thought. And in neurology, we don't have the kind of measurements that we would like to have, and they're not that feasible right now. I want to get back, though, to the question that George asked, which is, the question about surrogate biomarker. I do believe it's very complex. I believe that it could be that a surrogate biomarker is appropriate for an early stage of disease, but it may no longer be appropriate for later stages. So I believe a beta or amyloid is good for early stages, but I suspect you reach a stage of Alzheimer's where other processes intervene and are more responsible for progression. So I suspect that amyloid plaque lowering is not going to be a great biomarker for later stages of Alzheimer's, maybe even better for earlier stages, pre-symptomatic. I would also say that the degree of the effect on biomarker may also be very important. That's what I was trying to say, that having a small decrease in an amyloid plaque may not have very much clinical meaningfulness. And finally, and I hate to even bring this up, is the fact that these diseases are actually not just one disease. There are probably actually many diseases that we call Alzheimer's or Parkinson's, but there are already, we see, for example, some differences between ApoE4 carriers and non-carriers, not only in the use of these biomarkers as diagnostics, but probably also in the effect size that we see from these various drugs. And that's just the tip of the iceberg, I believe. So the question on whether or not a surrogate biomarker is appropriate is a very complex question. At the end of the day, it is a judgment call, and it's going to depend on all these complexities I just outlined. And there may be even more that I haven't even talked about.
I just have one quick comment I just want to make to Dr. Califf, since he's here too. When FDA put out its early AD guidance, we had submitted some comments with a number of other organizations and researchers to ask the agency to be more specific about measures for clinical meaningfulness. And I just want to reemphasize that the CDR sum of boxes was mentioned in that particular guidance, but nothing else was. And we, asked that more be mentioned. And I think that that's a role that the agency can play, is to put out more messaging to industry on what they can use and what you would like to see.
Just, I feel like I need to remind everybody I'm not at the FDA now, and I won't be unless the Senate confirms, which is still up for senatorial judgment.
Mm-hmm. Mm-hmm. Okay. So maybe we can just follow up a bit, because it seems clear from a number of studies and things that have been said on the panel today that the earlier one can intervene, the more likely one is to see an effect. As very early on, reduction or elimination of plaques could have a big effect, where later on it doesn't. So given that the studies are done in symptomatic patients, where are the symptomatic patients on that gradation of whether or not it's too late or isn't too late?
It's almost too late. I think it is, because that's why the effect sizes are pretty small, I believe. Mm-hmm. I believe that, but we know that by the time people have MCI, even mild cognitive impairment, the disease has been in progress for 20 years, and that the amyloid plaque burden has reached an asymptote, has reached a maximum. It actually doesn't grow very much, the burden, beyond after MCI. So I believe that it's almost too late, and that's why we have the effect sizes we see. That's my opinion.
I agree, Al, and I think it's reasonable to expect from this class of drugs in this stage of disease sort of 20 to 30% slowing of disease course, not 100%. That we could get much higher earlier in the disease. I think using symptoms to mark a certain stage of disease is extremely crude. We would never do that in cancer. So we try and stage the disease using pathologic biomarkers. So in this case, tau staging. And what we saw even in our phase 2 trial, and these are small samples for sure, is that patients, even who had about the same symptomatic stage of disease, people who had less tau in their brain were better responders to therapy, which is exactly what you would expect for any disease-modifying drug. It's just further evidence that everything in this theory is holding together, and I think gives us great hope that going earlier, either taking people who are mildly symptoms but have the earliest pathology, or people who have early pathology, no symptoms, is the way to go.
I very much agree with that. I think that it depends on what the drug is targeting also in the disease. So if you had a drug that was maintaining synapses or neuronal health, that drug may in fact have a big effect. We know that cholinesterase inhibitors, the target neurotransmitters, have an effect actually in more advanced symptomatic disease and mild dementia. So it really depends on the target. But for amyloid beta, based on how we understand the disease cascade right now, it certainly makes sense that the earlier you treat, the better. On the other hand, we talked about the difficulty of measuring changes in cognition in early symptomatic patients. Now, how do you determine what are clinically meaningful outcomes in people who have no symptoms at all, but are already showing brain changes consistent with Alzheimer's disease? That's another big challenge, I think. And there, I think surrogate biomarkers, establishing surrogate biomarkers in symptomatic disease and then applying them to preclinical disease may be the way we need to go in order to judge what these drugs are doing.
So Dan, you talked about a trial in patients with plaques but no symptoms. And so what are the endpoints of that trial, and how long is that trial going to take to complete?
Yeah, so we maybe took a page out of some cardiovascular research here. So it's an outcome-based trial where we're actually looking at events. So it depends on the event rate in the treatment group and the placebo group. The event is sort of progression to symptomatic disease, I guess you could say. I don't know how long it's going to take to enroll the study, first of all, which is its own little challenge to convince people to get a PET scan or in this case, a blood test to see what the pathology is, and then try an experimental therapy for something that's not causing them symptoms yet. But again, that's been done in prevention studies in cardiovascular disease, and then follow them to an outcome. But I'm guessing we can enroll it in about a year. And I hope 3 or 4 years after that, we'll have enough events, conversion to symptomatic disease, that we can unblind the study and see the results. So it's in the time frame of Alzheimer's research, which is, we're decades and decades into this. It's the near future.
Mm-hmm. So one question for Mark, because you talked about the $3,000 to $8,000 benefit. And how do you think about alleviation of suffering? I mean, it is such a devastating disease for patients that you can't measure the value of a drug just by what healthcare costs it saves. That there is some value to actually economic value if you don't have to care for someone at home and you can keep working. But there's also just value because people are suffering less, and that's worth something. So how do you think about assigning those kind of aspects?
No, I think that's a great question. And that is incorporated in those estimates. That the health benefits are, they're trying to capture those health benefits in addition to any savings from reductions in the need for other medical care. So I think in general, in the healthcare sector, we tend to look for what insurers and patients and physicians are willing to pay for things as a measure. But absolutely, the alleviation of suffering, the ability to have a better life, for sure, it's hugely valuable. And I think I think it's really challenging. It's uniquely challenging in this area relative to an area like we were talking about, cardiovascular or cancer, where the big effects are on mortality or hospitalizations. This is just a harder one, I think, to come up with what's it worth? Is it priceless? At some level, it's priceless, right? To have someone be able to live a few more years. I had a close family member pass away recently from Alzheimer's. It was devastating for all of us, for my family member and everything else. And, at some level, it feels priceless if she had been able to have a better life. So I think it's just a thorny area because I just, at a high level, I think about Medicare as a program. And Medicare is a program that I've studied a lot, and I'm thinking about the changing demographics in the US, and I'm an economist. And so I tend to think about, how does it all add up? So for my family member at Alzheimer's, I'd be willing to drain my 401k and my kids' college fund and everything else to have them have a better, but putting a dollar value on this for society when we have to make hard choices about resource allocation. And I think in this country, we're going to have to, this is like a microcosm of a bigger challenge that I think the country is going to be confronting increasingly as the demographics change. And as we, and so I just think it's a fascinating area. I'm honored to be a part of this. But I think I think it's worth trying to get a value of what people are willing to pay for the health increment to them or their family members, to figure out what is the right price. What is the Goldilocks just right price? The right price to induce innovation on the one hand, but preserve access and not bankrupt programs. And I agree with Sue, by the way. She made this point that people had sort of gone a little too much to sort of talk about, oh my gosh, this is going to cost so much money. If everybody with Alzheimer's gets this drug, what's it going to cost? And that was probably not helpful to the national dialogue on this.
So I know that we could keep going all day, but I have the Oscars music they're playing us off the screen. So I'd like to thank all of the panelists. It's really been a very engaging discussion, and hand it back over now to Kathy to introduce the next speaker. Thank you.